RNA Isolation Protocol. Total RNA isolation from bacterial cells. Principle of RNA isolation. Total RNA is isolated and separated from DNA and protein after extraction with a solution called as trizol. Trizol is an acidic solution containing guanidinium thiocyanate, GITC, phenol, and chloroform. GITC irreversibly denatures proteins and RNases. This is followed by centrifugation. Under acidic conditions, total RNA remains in the upper aqueous phase, while most of DNA and proteins remain either in the interphase or in the lower organic phase. Total RNA is then recovered by precipitation with isopropanol. RNase enzymes can be inactivated by including diethyl pyrocarbonate, DPC. Materials required for RNA isolation. Bacterial culture. Trizol. Chloroform. Isopropanol solution. TAE buffer. 70% ethanol. Procedure of RNA isolation. Take 800 ml of bacterial culture in a fresh Eppendorf. To this add 160 ml of trizol, 1/5 of culture volume. The solution was mixed well by pipetting several times. To this add 32 ml of chloroform, 1/5 volume of trizol. Incubate for 2 to 5 minutes and centrifuge at 12,000 rpm for 15 minutes at 4 degrees Celsius. Transfer the aqueous phase into a new tube and add equal volume of isopropanol. Mix well. Centrifuge at 10,000 rpm for 10 minutes at 4 degrees Celsius. Discard the supernatant and resuspend the pellet in 70% ethanol. Again centrifuge at 10,000 rpm for 10 minutes at 4 degrees Celsius. Discard the supernatant. Air dry the pellet at 37 degrees Celsius for 10 to 15 minutes. Resuspend the pellet in 50 ml of TE buffer. Analyze the RNA sample, quantitatively and qualitatively. Isolation of RNA from animal cells or tissues. Materials reagents. Tiorizol reagent. Chloroform. Instruments. Fume hood. Vortexer. Micropipettes. Chilled microcentrifuge. Pellet pestle homogenizer. Clean the workstation, pipetter shaft, bench top, with a surface decontamination solution that destroys RNases. Chill the microcentrifuge to 4 degrees Celsius. Homogenization fresh tissue or cell culture. Fresh tissue is preferable for optimal RNA isolation. Alternatively, tissue should be submerged in RNA stabilization reagent immediately after dissection and stored at -80 degrees Celsius. Add 1 ml tiorizol reagent per 100 mg fresh tissue, mince on ice using sterile scalpels, and homogenize with a sterile pellet pestle probe. In case of cell cultures, they should be processed immediately after removal from the incubator. Either centrifuge cells grown in suspension at 300 Xg for 5 minutes at RT, 15 to 25 degrees Celsius, and discard supernatant or remove the culture medium from cells grown in monolayer. Add 1 ml tiorizol reagent per 1/107 cells to cell pellets or directly to the culture dish or flask for cells grown in monolayer. Resuspend the lysate with a sterile, disposable 1 ml pipette tip. Transfer the tissue or cell lysate to a 2 ml siliconized low-retention tube. Pass sample through sterile, disposable 21G needle 10 times. In doing so, high molecular weight cellular components, DNA, are fragmented, thus minimizing their presence in the aqueous phase. Let homogenate sit at RT, 15 to 25 degrees Celsius, for 5 minutes for complete dissociation of nucleoprotein complexes. Let homogenate sit at RT, 15 to 25 degrees Celsius, for 3 minutes. Centrifuge at 12,000 Xg for 15 minutes at 4 degrees Celsius. After centrifugation, heat the centrifuge to room temperature, 15 to 25 degrees Celsius. Transfer upper aqueous phase 600 ul to new 1.5 ml RNase-free tube. Proceed to alcohol precipitation. Precipitate the RNA from the aqueous phase by mixing with isopropyl alcohol. Use 0.5 ml of isopropyl alcohol per 1 ml of trisol reagent used for the initial homogenization. Incubate samples at 15 to 30 OC for 10 minutes and centrifuge at not more than 12,000 Xg for 10 minutes at 2 to 4 OC. Remove the supernatant completely. Wash the RNA pellet once with 75% ethanol, adding at least 1 ml of 75% ethanol per 1 ml of trisol reagent used for the initial homogenization. Mix the samples by vortexing and centrifuge at no more than 7,500 Xg for 5 minutes at 2 to 8 OC. Repeat. Air dry or vacuum dry RNA pellet for 5 to 10 minutes. Dissolve RNA in DPC treated water by passing solution a few times through a pipette tip. Subject to spectrophotometric analysis to determine sample concentration and purity. Expected results. A gel-like pellet on the side and bottom of the tube of RNA precipitate. On taking OD at 260 nanometers and 280 nanometers, the A260/A280 ratio should be above 1.6.